Hey friends, in this session we would be revising physiology and nerve and muscle and digestive system. Okay. Molecular basis of contraction. At rest, myosin heads are bound to ADP and said to be in cocked position in relation to actin. On response, calcium bind to troponin tropomyosin complex induces a conformational change in this filament that allows for myosin head to cross a bridge with actin. Myosin head rotate, move actin and shorten muscle fiber forming power stroke. At end of power stroke, ATP binds to now exposed site and causes detachment. ATP is hydrolyzed into ADP and PI and this recock the myosin head. Cycle repeats as long as calcium and ATP are present. Each power stroke shortens the sarcomere by 10 nanometer. Myosin head hydrolyzes ATP and becomes reoriented and energized. Myosin head binds to actin forming cross bridges. Myosin head rotates towards center of the sarcomere power stroke. As myosin heads bind ATP, the cross bridges detached from each. Myosin head hydrolyzes ATP and becomes reoriented and energized. Contraction cycle continues if ATP is available and calcium level in the sarcoplasm is high. Excitation contraction coupling process by which membrane depolarization of muscle fiber initiate contraction process by which membrane depolarization of muscle fiber initiate contraction. This is called excitation contraction coupling. Steps are discharge of motor neuron, release of acetylcholine, binding of acetylcholine, sodium and potassium conduction increases, generation of action potential, inward spread of depolarization along T-tubule, release of calcium from sarcoplasmic reticulum, binding of calcium to troponin C, formation of cross bridge and muscle contraction. So this is about excitation contraction coupling, the process by which membrane depolarization of muscle fiber initiates contraction. Discharge of motor neuron, release of acetylcholine, binding of acetylcholine, sodium and potassium conduction increases, generation of action potential, inward spread of depolarizing along T-tubule, release of calcium from sarcoplasmic reticulum, binding of calcium to troponin C, release of cross linkages, muscle contracts. Calcium pumped back, release of calcium from troponin, cessation of interaction between actin and myosin, and muscle relax. Neurotrans Neuromuscular junction sequence of events. It is junction between terminal branch of nerve fiber and muscle fiber. Axon, vesicles containing neurotransmitters, mitochondria, synaptic cleft, postsynaptic membrane. Opening of voltage gated calcium channel, entry of calcium ions from extracellular fluid, opening of vesicles and release of acetylcholine, passage of acetylcholine by a synaptic cleft, binding of acetylcholine with receptors and formation of acetylcholine receptor complex, opening of ligand gated calcium channels, entry of sodium from ECF, action potential generation, excitation contraction coupling, muscle contraction. Layers of alimentary canal. Typical cross section of interstitial wall includes following layer from outside surface inwards: the serosa, the longitudinal muscle layer, circular muscle layer, submucosa and mucosa. Serosa, longitudinal muscle, circular muscle, submucosa, mucosal muscle, mucosa, epithelium lining, mucosal gland, myenteric nerve plexus, deficiency of which causes aganglionic megacolon, and Meissner nerve plexus, also called submucosal plexus, and Meinderic nerve plexus is Orbach plexus, submucosal gland, and mesentery, saliva, and its secretions. Basic mechanism: contents of food with epithelium stimulate secretions. Type of stimuli that activates these systems are tactile stimulation, chemical irritation, or distension of gut wall. Autonomic stimulation. Parasympathetic stimulation increases the alimentary tract and gallbladder rate. Sympathetic stimulation has dual effect. Alone, it usually increases secretion. If parasympathetic is already causing secretion, its stimulation usually reduces the secretion. By hormones, several hormones regulate the volume and character of secretions. They are usually polypeptides or its derivatives. Basic mechanism of secretion by glandular cells. Nutrient materials needed for secretion must defense or actively transported by blood into base of glandular cells. Energy, ATP produced by mitochondria along with appropriate substrate is then used to synthesize secreting substance entirely in endoplasmic reticulum and Golgi complexes, ribosomes for proteins. Secretory substances from tubules of endoplasmic reticulum, vesicles of Golgi complexes. In Golgi bodies, material are modified, concentrated, and discharged into cytoplasm as secretory vesicles. Vesicles remain stored until nervous or hormonal control signals causes the cell to extrude the vascular contents. Different salivary glands. Primary glands are parotid, only serous, submandibular and sublingual, both serous and mucous. Tiny glands are buccal, labial and glossopalatine, only mucus. Daily secretion is 800 to 1500 ml, approximately 1000 ml. Saliva contains two major types of protein secretions. Serous secretion contains ptyalin and mucus secretion mucin for lubricating and protection. pH is 6 to 7 or you can say 6.8, which is favorable range for ptyalin. Composition of saliva contains large quantities of K plus and bicarbonate ions. Sodium and chloride are several times less in saliva than in plasma. Composition: water and solutes. Solutes are inorganic and organic. Inorganic are sodium chloride, Na plus, Cl minus, K plus, bicarbonate, calcium, PO4 3 minus, thiocyanate, SCN minus. And organic are ptyalin, mucin, proline-rich protein IgA, lysozyme and lipase, peroxidase, statins, statherine, calcarine and lactoferrin, IgA, mucin, ptyalin, Na plus, Cl minus, K plus, bicarbonate, calcium, PO4 3 minus and thiocyanate. Mechanism of secretion: first stage. Acini secretes a primary secretion, saliva, Cl out and Na out and K plus in. In saliva, Na and chloride 15 milliequivalent per liter, 1/10 of plasma. K plus 30 milliequivalent per liter, 7 times as in plasma. Bicarbonate 50 to 70 milliequivalent per liter, 2 to 3 times as in plasma, higher. During maximal salivation, the vary ionic concentration change considerably because rate of formation of primary secretion by acini can increase by 20 folds. Thus, ionic content balance decreases in pieces and striated and excretory ducts. Add sodium, only one half or two-thirds of plasma, and potassium rise only to 4 times. Contains amylase, sodium potassium chloride bicarbonate similar to plasma. Functions of saliva: defense, antibacterial, antifungal, antiviral, immunological, IgA, thiocyanate, lysozymes, proteolytic enzymes. Digestive functions: styline, lipase, formation of bolus, taste. Protective functions: coating for soft tissues, for hard tissue, teeth, lubrication, keeps oral cavity moist, facilitates speech, mastication and swallowing, and buffering. Defense, digestive functions, protective functions, lubrication, buffering. Neuronal regulation, mainly controlled by parasympathetic nervous signals all the way from superior and inferior salivary nucleus in brainstem, located at junctions of medulla and pons 6, 7, 8 pontomedullary junctions. CNS, tactile and taste stimulation, superior and inferior salivary nuclei, salivary glands, secretion or inhibition. Vasodilation by parasympathetic, increased salivary production. Calcarein secreted by salivary cells, which in turn act as an enzyme to split one of the blood proteins, and alpha-2 globulin to form bradykinin, a strong vasodilator. Bradykinin is a strong vasodilator, in turn increases salivary secretion. Superior and inferior salivary nucleus, tractus solitarius, glossopharyngeal nerve, facial nerve. As you know that glossopharyngeal nerves, tongue, otic ganglion, and parotid gland by via auriculotympanic ATN, auriculotympanic nerve, and facial nerves, chora tympani, submandibular ganglion, submandibular gland, and sublingual gland. Superior salivary nucleus and inferior salivary nucleus. Daily secretions of intestinal juice: saliva 1000 ml, pH 6 to 7, gastric secretion 1500 ml, pH is 1 to 3.5, pancreatic secretion is 1000 ml, pH is 8 to 8.3, and bile is 1000 ml, 7.8. Small intestinal secretion 1800 ml and 7.5 to 8. Brunner's gland secretion 200 and pH is 8 to 8.9. Large intestinal secretions 200 and pH is 7.5 to 8. Total is 6700. Gastric secretion: action and peptic ulcer. Gastric secretion. Types of gland: oxantic gland, gastric gland. Oxantic or parietal gland secretes HCl and intrinsic factor, and chief cells or peptic cells secretes pepsinogen. Intrinsic factor is produced by oxantic cells and mucus located on inside surface of body and fundus proximal 80%. Secretions are mucus, neck cells, mucus, peptic or chief cells, pepsinogen, parietal or oxantic cell, HCl, and intrinsic factor. Surface epithelium, mucus, neck cell, oxantic or parietal cell, peptic or chief cells. Pyloric gland mainly secretes mucus, also gastrin, located in antral portion of stomach, distal 20%. Type of cell present is mucus neck cell producing mucus, peptic cell pepsinogen, and G cell gastrin. Mucus neck cells, oxantic or parietal cells, canaliculi, and secretion. Surface mucus cell, entire surface of stomach, mucosa between glands has a continuous layer of a special type of mucous cells called surface mucous cells. Gastric anatomy, anatomical region of stomach, fundus, cardiac, body, pyloric antrum. Sections of gastric wall, gastric pit, mucosa, outer muscular layer, inner muscular layer, submucosa, muscularis mucosa, mucosa, gastric pit, secretion of gastric gland. Section of gastric or oxantic mucosa. Oxantic gland supplied with extensive net of fenestrated capillaries that drain into subepithelium plexus. Submucosal artery, submucosal vein, submuscularis mucosa, glandular epithelium, capillary around oxantic gland, central venule, subepithelium, capillary plexus, gastric pit, surface epithelium. Surface mucous cells. Entire surface of stomach mucus between glands has a continuous layer of a special type of mucous cells called surface mucous cells. Coat stomach surface by 1 mm thick gel-like viscous mucus. Mucus is alkaline and protects from acidic proteolytic stomach secretion. Composition of gastric secretion. Composition: water solutes, inorganic and organic. Amount is 1.5 to 2 liters. Inorganic HCl, sodium, potassium and bicarbonate, and organic enzymes like pepsin, gastric lipase, gelatinase, intrinsic factor. Mechanism of HCl secretion. Parietal cell secretes an acid solution that contains 160 millimole per liter of HCl. pH of acid 0.8 at first, H plus concentration at this pH 3 million times than that of blood. About 1500 cal of energy is required per liter of gastric juice. The main driving force is H plus K plus ATPase pump. This is very important. Interstitial fluid. So firstly what happens is CO2 combines with water to form bicarb H2CO3 and which splits into H plus and HCO3 minus. HCO3 minus and, and chloride antipode increases bicarbonate level in blood, so alkaline tide occurs after eating food. So this H plus combines with that chloride which comes into the lumen side and combines with H plus, and this all H plus is transported to the lumen side via the help of H plus K plus ATPase, and H plus and Cl minus combine to form HCl. H plus HCl 150 to 160 milliequivalent per liter, KCl 15 to 60 milliequivalent per liter, small amount of NaCl. Mucous neck cells, oxantic or parietal cells, canaliculi and secretion. HCl is formed at the villus-like projections inside the canaliculi and in the transported through the canaliculi to secretory end of the cell. Regulation of gastric acid secretion. Three main there are main factors are acetylcholine, gastrin and histamine. Acetylcholine through M3 muscarinic receptor, gastrin and histamine through H2 receptors. Parasympathetic G cells through blood, gastrin, ECL, acetylcholine, histamine, gastrin, adenyl cyclase, histamine C, AMP, gastrin, calcium, and parietal cells, calcium, lumen, H plus K plus ADPase. So here you can see H plus K plus ADPase is important. Acetylcholine by parasympathetic stimulates HCl secretions by parietal cells, also pepsinogen from peptic cells. Histamine by enterochromaffin-like cells stimulates HCl secretions. ECL itself is stimulated by gastrin. Gastrin by gastric cells, G cells, large polypeptide, stimulates the HCl secretions. Phase of gastric secretion is cephalic, gastric, and intestinal phase, CGI. Like you can learn it by Chief Justice of India, CJI. So just convert that J from to G. CGI, cephalic, gastric, and intestinal phase. Phases of gastric secretion. Cephalic phase occurs even before food enters the stomach, especially when it is being eaten. It results from sight, smell, thought, or taste of food, and the greater appetite, the more intense is the stimulation. This phase of secretion normally accounts for about 30% of the gastric secretion associated with eating a meal. Regulation. Neurogenic signals that cause the cephalic phase originates in the cerebral cortex and in the appetite centers of the amygdala and hypothalamus. They are transmitted through dorsal motor nuclei of the vagus and through the vagus nerve to the stomach. Gastric phase occurs once food enters the stomach. When it turns, causes secretion of gastric juice through several hours while food remains in the stomach. The gastric phase of secretion accounts for about 60% of the total gastric secretion associated with eating a meal. This phase is regulated by long vasovagal reflexes from stomach to the brain and back to the stomach. Local enteric reflex, the gastric histamine mechanism. Intestinal phase. When food enters the upper portion of the small intestine, particularly in duodenum, small amounts of gastric juice continue to be secreted. This phase accounts for about 10% of acid response to a meal. So mostly gastric phase, then cephalic phase, and then intestinal phase contributes. This phase is regulated by nervous mechanism, hormonal mechanism, probably partially because of small amounts of gastrin released by the duodenal mucosa. Inhibition of gastric secretion. Formation of chyme inhibits gastric secretion by reverse enterogastric reflex. Mesenteric nervous system, extrinsic, sympathetic, and vagus inhibition of gastric secretion. Presence of acid, fat, protein breakdown products in upper small intestine. Secretion inhibits gastric secretion. Vagal center of medulla. Afferent fibers, food secretory fiber, vagus trunk, local nervous plexus, cephalic phase via vagus, parasympathetic excites pepsin and acid production, gastric phase, local nervous secretory reflexes, vagal reflexes, and gastric histamine stimulation, circulatory system, intestinal phase, nervous mechanism and hormonal mechanisms. Other enzymes, glucose-dependent insulinotropic peptide, gastrin-inhibitory peptide, vasoactive intestinal peptide, somatostatin, all through inhibits gastric secretion. Somatostatin, VIP and GIP. GIP, VIP and somatostatin inhibits gastric secretion. Gastrointestinal hormones are major GI hormones: gastrin, secretin, cholecystokinin, gastric inhibitory peptide, mortalin, and peptide YY. Gastrin, G cells in antrum, stomach, and duodenum. Stretch peptides and amino acids and vagus primary stimuli, and general actions is increase gastric H plus, gastric mixing, lower GI tract motility. Secretin, S cells of duodenum. Primary stimuli is acidic chyme. Increase pancreatic buffer bicarbonate secretion, increase biliary and small intestinal buffer secretion, decrease gastric H plus by gastrin, decrease gastric emptying, cholecystokinin, I cells of duodenum and jejunum. Primary stimuli for CCK is small peptides and amino acids and fats. Pancreatic enzyme secretion increases contraction of gallbladder and relaxes sphincter of Oddi, increases pancreatic and biliary buffer secretion, decreases gastric emptying, increases lower GI tract motility. Gastric inhibitory peptide or GIP, duodenum and jejunum fatty acids, glucose and amino acids, decreases gastric H plus secretion, increases pancreatic insulin secretion, decreases gastric emptying. Motilene, MO cells of duodenum. Primary stimuli is fasting, increases phase 3 contraction of the MMC, peptide YYA, L cells of intestine. MMC is for migratory motor complex. Primary stimuli is fatty acid, glucose, hydrolyzes protein, inhibition of gastric emptying, pancreatic secretion, gastric acid secretion, intestinal motility and food intake. Peptic ulcer. High acid and pepsin content, irritation, poor blood supply, poor secretion of mucus, infection, H. pylori, pylorus, ulcer sites, cardiac, marginal ulcer and pylorus. Specific causes: bacterium Helicobacter pylori breaks down the gastrointestinal mucosal barrier and stimulates gastric acid secretion. Other causes: smoking, consumption of alcohol, consumption of aspirin and other non-steroidal anti-inflammatory drug, NSAIDs. 2. Mechanism for neutralization of excess acid: nervous reflexes and hormonal feedback from duodenum. Presence of acid in duodenum liberates secretin from intestinal mucosa. Pancreas stimulates and secretes bicarbonate for neutralization. Rentidine, cimetidine. Treatment of peptic ulcer. Since the discovery that much peptic ulceration has an infectious basis by the bacterium H. pylori, therapy has changed immensely. Almost all patients with peptic ulcerations can be treated effectively by two measures: use of antibiotics along with other agents to kill infectious bacteria, administration of an acid suppressant drug such as rentidine and similar newer molecules, which are antihistaminic agents that block the stimulatory effect of histamine on gastric glands histamine receptors, that reduces gastric acid secretion by 70 to 80%. More recently, proton pump inhibitors such as omeprazole and similar newer molecules are used to suppress acid production. Other measures that can be used to treat peptic ulcers includes the following: neutralize the gastric acids with antacids. However, this method is not used recently. Bismuth compounds are used that probably enhance the mucosal barrier and are anti-secretory. Cryoprotective agents such as sucralfate and an aluminum salt of sucrose octasulfate appears to help with the healing of ulcer, probably by forming a protective coating. Prostaglandin analogs such as misoprostol can be used to reduce the incidence of peptic ulcers in patients who are on long-term NSAID treatment. Other conservative measures such as quitting smoking, reducing in caffeine and alcohol consumption, and measures to reduce stress can speed up the healing process and prevent ulcers from recurring. Biliary system.